Okay, good afternoon. My name is Charlie Simpson, and I am Executive Director of Marketing Communications for the Carle, Illinois College of Medicine. Welcome, everybody. I'm going to give a brief introduction about Carle, Illinois, and then I'll introduce our first speaker. So the mission of the Carle, Illinois College of Medicine is to educate exceptional physician innovators to deliver high-value, compassionate healthcare through transformative solutions developed at the intersection of engineering, science, and medicine. The Carle, Illinois community is driven by 4 key values that really permeate our culture and drive our curriculum. And we refer to them as the 4 C's. They're compassion, competence, curiosity, and creativity. And we're committed to creating a diverse and talented community of students, faculty, and staff. And that's really essential to fulfilling our mission. We actively promote an inclusive environment in which our students, faculty, and staff can learn, teach, reserve, and serve. And this includes supporting and providing opportunities for those that are underrepresented in medicine by having programs such as this REACH program. So if you don't know, REACH stands for Research and Education for the Advancement of Compassionate Healthcare. And it's an intensive clinical and research immersion summer experience for undergraduate students interested in pursuing a career as a physician. In addition to helping students prepare for med school, REACH was developed to grow the presence of underrepresented minorities in the field of medicine overall. So you can learn more about the Carle, Illinois College of Medicine by visiting our website, and you can find the REACH information under the Research and Innovation tab. So that's at medicine.illinois.edu. When you go there, you can sign up to be notified of any future information or opportunities about REACH. And you can also email us anytime you have any questions or need more information at reach@medicine.illinois.edu. So with that, I'm going to introduce our speaker. Dr. Marty Burke is the May and Ving Lee Professor for Chemical Innovation and Associate Dean for Research here at the Carle, Illinois College of Medicine. Dr. Burke's group has pioneered the field of molecular prosthetics, including the development of an automated Lego-like platform for democratizing small molecule synthesis. Dr. Burke has received a number of honors and awards, including the Nobel Laureate Signature Award for Graduate Education in Chemistry from the American Chemical Society. And he's garnered praise for excellence in teaching at both the undergraduate and graduate levels. Dr. Burke is an academic co-founder of Revolution Medicines and founder of Sfunga Therapeutics, and is currently leading a team deploying scalable COVID-19 testing on campus. So with that, I'm going to turn over to Dr. Burke.
Thank you so much, Charlie, and great to have the chance to speak with all of you. Thank you so much for tuning in. We couldn't be more excited to have the chance to talk with you today. I'm going to try to share my screen to make sure this works. And Charlie, are we able to see my screen now?
Yes, perfectly.
Okay, great. So thanks to all of you so much again for tuning in. My name is Marty Burke, and I'm a researcher myself, as well as the Associate Dean for Research at the College of Medicine. And I'm really excited to have the chance today to tell you just a little bit about some of the things that my students are working on in our lab, as well as a campus-wide effort to try to help defend our campus community against the really challenging problem associated with COVID-19. The title is Making Molecular Prosthetics, which is a field that my group is really excited to be working in this space, but I'll also talk about a bunch of other cool molecular stuff that we're really fired up about trying to gain access to. And the goal of the group in large part is to try to make it easier for everybody to make molecules, because we think, what an amazing world it would be if it was easy for everyone to participate in this process, and all those really cool ideas we're probably missing out on because currently there's barriers that exist in terms of the kind of road that you have to travel down to be able to participate in this process. And we'd love to shatter those barriers and make it a lot easier for everybody to participate. Okay, so this is one of our big objectives. Okay, so jumping right in. So when I was about your age, I did one of those MD PhD things. So kind of running back and forth between the hospital and the organic chemistry lab, trying to think and dream about problems that would love to tackle as a researcher. And one of the things that really knocked my socks off, honestly, as a medical student is seeing the power of small molecules to help people get better. So I know this sounds kind of obvious probably because we're all familiar with this idea of medicines and that they can help us heal, but it's actually an amazing thing to see it. You know, we have a patient who's sick and you give them a molecule and then watch them get better. This really kind of blew my mind, honestly, how powerful that was as a vehicle for positively impacting health. And many of you have heard of some of these molecules, things like penicillin and Lipitor, that many of ourselves or even our parents have probably taken. Penicillin to treat infections and Lipitor to help treat high levels of cholesterol, for example. And these medicines all work kind of the same way, in which they usually bind to a protein, especially if that protein is doing something that's not good for us. So kind of a hyperactive protein that's causing problems. So these small molecules bind to the protein and they turn the protein off. And this is very powerful. If you're sick because of too much function, then this kind of turn off mechanism can really help make a difference. The thing that struck me, however, was that if you're sick, not because of too much function, but if instead you're sick because you're missing something, you have a lack of function that's causing you to be ill, then actually this whole paradigm falls apart, because there is no protein to turn off because you're actually lacking function in the first place. And it was actually a young woman that I met when I was on my pediatrics rotations that really kind of helped me realize how powerful this problem was. So she had cystic fibrosis, and so this is a disease in the lungs where you're missing a protein called the cystic fibrosis transmembrane conductance regulator. It's a very long word. We call it CFTR. It's a protein that transports anions across the epithelium of your lung. And if you're missing this protein, you can't fight infections in your lung. And so these young people actually end up struggling with lung infections throughout their whole life. And unfortunately, it shortens their lifespan substantially. And so I was explaining to this young woman, she's like a brilliant college student, and she's super sharp. And I was telling her all about the deficiency of CFTR and how that was causing her trouble. And she said to me, she said, "Look, it sounds like you know exactly what's wrong with me. Why can't you fix it?" So it was one of those very powerful kind of conversations, and boy, she was right. She put her finger on a critically important gap where we have a fundamental understanding of the missing link, and yet the only thing that we could offer her was things to treat the symptoms. There was nothing to actually address the fundamental defect. And so that conversation, powerfully challenged me to think about, why can't we fix this problem? And, she really kind of inspired me to think about maybe this whole small molecule drug thing. Could we bring that to bear upon this loss of function problem? And the idea that emerged that I kind of got really excited about, could we develop molecules that could do what this protein is supposed to be doing? Almost like a prosthesis on the molecular scale, right? So if you're missing a hand, an artificial prosthetic hand can actually give you a lot of positive impact. Could we develop, almost like artificial ion channels that are molecule based that could replace her missing protein? So this concept, once I kind of got really excited about it, sometimes you keep thinking about something, and the more you think about it, the more excited you get. Well, that's kind of what happened for me here. So we started imagining maybe this could apply to lots of other types of diseases, like if you're missing enzymes or proteins that transport oxygen in your blood, like hemoglobin, and lots of others. And so could we develop a field of medicine where small molecules replace missing proteins and thereby help people become healthy when you're missing a function? And so when I had the chance to start my own research group here at the University of Illinois, we went all in on this problem, and I was able to recruit a bunch of graduate students, and we launched a new lab, and this was the problem that we decided to try to address. So it was very interesting because when we started talking about this, there was a lot of people telling us this wasn't going to work. And in fact, there were people who told me I was throwing my career away because this was not only going to fail, but I was going to lose my chance to do something else that might work. And so this was a really bad idea. I tell that story because I think it's common that oftentimes when you get really excited about something, you might be the only person who thinks it's a good idea, but that's okay. Like, it's good to listen to people's criticism, but if your gut instincts are telling you, I really think this might be worth pursuing, sometimes you kind of have to just go with your gut. And worst case scenario, you're wrong, and then you find something else to work on. So I was lucky to have students who were excited about this with me, and we just went for it. And the argument why this wasn't going to work is that because these proteins are really complex and very sophisticated and very specialized, and they do what they're supposed to do very effectively. And so the argument was, you're never going to perfectly replace one of these proteins with a small molecule drug, and therefore it's not going to be effective. So our kind of different way of thinking about this was based on what we call this robustness hypothesis. So it turns out that all of us at the molecular level, we're very robust. What that means is we have a lot more function than we need to do most of the things that are important. And the evidence for this comes actually from a lot of really cool studies in human genomics. So there was a recent study out of the Broad Institute at Harvard MIT where they took a bunch of healthy people, they sequenced their entire genome, and they found that on average, we're each missing about 100 proteins. Okay, so we have 100 missing functions, and yet we're okay, or at least we think we're okay. And the idea is you can lose a lot of things and still be okay because you have a lot more than you need to be functional. So we have an inherent level of excess function that allows us to tolerate loss of function in most cases. It's only in this kind of rare case where if you're missing one protein, you drop below some threshold of required function, and now you've got a disease because you're not at the level that you need for physiology. But you haven't dropped to zero. And this was kind of our idea, is that because of your inherent robustness, all of your backup mechanisms and other proteins and everything you've got is trying to get back to healthy. You just can't quite get there, okay, because of this loss of this one protein. And so we thought, well, maybe that means you don't have to perfectly replace the missing protein to get back to healthy. You just need something that's good enough. Kind of like if you're losing a hand, even a pretty simple prosthetic device can be pretty effective because you get creative and your other hand works harder and you find ways to solve problems. And so we said, if you had a small molecule that could even partially replicate the function of that missing protein, perhaps that imperfect molecular prosthetic would be sufficient to get above this threshold, and now you get back to being healthy, okay? And if that turned out to be true, then all those naysayers might actually realize, wow, this might work because imperfection is sufficient. And if imperfection is sufficient, then all of a sudden we've got a powerful mechanism to try to help make a difference, okay? So this was really the key hypothesis that we were trying to test. Imperfect molecular prostheses could be sufficient to restore health when you're missing a protein. And I'm excited to tell you now, 15 years later, we've got lots of examples showing that's true. And that has opened up a really exciting new field of medicine where we're going to try to do this in lots of different ways. So I'll tell you the story of how we got there. So here's one of the targets that we went after when we were getting started. I'll get back to the cystic fibrosis story in a second. There's another really important protein called ferroportin, which gets its name from ferro, which is the idea of a ferrous or iron. This goes back to the Greek word for iron, and portin, which is just transporter. Okay, so this is the iron transporter that you and I are using right now to move iron around in our bodies. And if you're missing this protein, you get anemia because you can't move the iron, and therefore you can't make your red blood cells. So there's a lot of people, millions of people in the world who have anemia because they're missing this protein. And so the idea was, if you have the protein, iron can flow through these lipid bilayers that are surrounding your cells. If you don't have this protein, it can't. And so we hypothesized then the iron would get built up and you would get these gradients that would form. And the gradients would form kind of precisely in the places where you're missing the protein. Okay, so kind of like a dam where water is building up behind it, there would be this powerful driving force for correction of this deficiency if we could find a small molecule that could bind the iron atoms and move them across the lipid bilayer. Then only in the places where you get this buildup of a gradient, you might get this correction effect. And so even if this molecule isn't perfect, meaning it might even bind other ions and it might not know it's supposed to move them in this direction, but because of the gradient that exists, the molecule would recover the required flow of iron atoms and thereby recover the physiology. Okay, so imperfection could be sufficient because the context in which this deficiency creates gives us a driving force, okay, for the iron atoms to move. Again, very much like water that's building up behind a dam, and if you poke a hole in the dam, the water is going to flow in the direction that it wanted to go in the first place, okay? So this was the idea, and now the challenge was finding a molecule, okay, that could do this. So, and I think when people tell these stories, then they say, oh, and then we went into lab and we found this molecule, as if that was easy. I'll tell you very honestly, that is not what happened. Okay, so we struggled. When I say we, this is myself, but also working very closely with my student Anthony Grillo, and several others in the lab. So I'll just tell you, we failed about 250 times. So we worked on this problem for about 3 years and failed and failed and failed and failed and failed. And it's to the credit of students like Tony and others in the lab, we just didn't give up. And I think one of the key messages I learned from this story is that it's really important to be able to fail. If you're trying to do something impactful, almost by definition, it's going to be really hard, and failure is an option, and it actually has to be in order for finally to break through. So I'd say we failed about 250 times, and then one day we didn't. So Tony had this amazing day in a lab where he'd used this assay based on yeast, which are kind of like us, but they're just obviously a lot smaller and a lot older in terms of evolution, but they're our eukaryotic cousins. They have a lot of the same hardwired machinery. And he had a yeast cell that was missing its iron transporter, and it couldn't grow. And he checked many, many different molecules in many, many different conditions, and he failed and he failed and he failed. And then one day we saw this. So this is a ring of cells growing around a paper disc that contains this molecule called hinokitiol. This is a molecule that's actually isolated from the bark of the hinoki tree in Taiwan. And it turns out it's an amazing natural product that wraps around an iron atom. And there's 3 molecules of hinokitiol that now form a shell around the iron atom. And it creates a greasy kind of ball that can go through the lipid membranes, and then it releases the iron on the other side. So this is the 3D printed version, okay, of this complex. You've got a green iron atom in the middle, and you can kind of see like the grease around it, and it allows it to go through the membrane. And then once it does, it just releases its iron on the other side. And it's like this really exciting, then amazing discovery for us that we were able to run with, okay? Turns out this molecule isn't perfect as a replacement for the protein. And we showed it wasn't perfect. It moves lots of other metals as well. But in the context of this protein deficiency, because of those gradients that are building up, we were able to show that only the iron gets corrected, okay? Everything else, you'll notice the gray bar and the blue bar are very much the same. Only for the iron, we saw this increase in flow. And this is really the first exciting evidence of this imperfect is enough, okay, to get us there. And this was really an exciting day in the lab. So just to kind of fast forward, Tony teamed up with Anna Santa Maria in my lab, and they made a fantastic team that went all in to try to figure this out. We showed in gut epithelia derived from people, so you can almost like grow little guts in the lab. It's actually kind of wild that this works. And you can take the proteins in your gut and knock them out, and then show that you lose the iron transport as indicated by this decrease going from this bar to this bar. And then we showed if we add our molecule, we got everything back. So back to normal in terms of the iron flow by adding this small molecule. We were able to do the same thing in red blood cells. So this is cells that are turning into the types of red blood cells that we use to carry iron in our body. And we showed that if you're missing the protein, you can't make red blood cells. But if we add the small molecule, you get recovery, and then they turn pink because they've become normal red blood cells. And we even showed with some dyes that you could see the iron getting trapped in certain parts of the cell. And then when we add our molecule, we can see it get released and watch this process happen. I think this was my favorite experiment from this whole project. There's these very cool animal models we use. They're called zebra fish. It's actually fish that you can see through them. So you can see a lot of the physiology with your eyes. And so this is a fish that's normal, and you can actually see their blood. Okay, so this brown part is the blood of the fish that's been stained. And while they're still alive, you can actually see their blood inside the fish. If we knock out their iron transporter, they cannot make blood. So they actually have a severely anemic fish, and you can see that because there's no blood to visualize. And then we simply added this molecule to the fish tank, and we saw for the first time in an animal recovery of an important physiological function with the small molecule replacing their missing protein. Okay, so this was the first time we ever, or anybody ever, saw molecular prosthetics in an animal. So we were really excited about that result, and this gave us a lot of momentum. We showed the same types of things can be done in mice, so we can recover the uptake of iron through their gut. we can also recover the formation of red blood cells. So we were able to increase the hematocrit of a mouse that was missing ferroportin, which really starts to connect to potential of treating people with the ferroportin deficiency. And so at this point, we were really excited. We spun out a company. So this is one of the cool mechanisms you can use to get research brought to society, is that there's a point where the lab has done a lot of what it can do, and now you got to kind of hand it off to a professional drug development group. So we started this company called Ambus Medicines. We got investment from investors in Boston, Massachusetts, and in San Francisco, and then we launched this company, and it's a very exciting story. There's now about 60 people working together in a lab out in San Francisco, and moving forward towards translating this into clinical trials to help treat people with missing proteins in their liver. Okay, so the next story I'll tell you is back to the cystic fibrosis. So as I mentioned, this was kind of a key takeoff point for us. We wanted to try to replace the missing CFTR protein that was missing in that young woman that I met during my pediatrics rotation. And it's a very interesting similar situation in which normally the CFTR sits at the top of your epithelia of your lung, and there's these anions like bicarbonate and chloride that are moving from the bottom of your epithelium and the blood that's flowing here to the apical side, or the outside that's facing into the air. And that helps protect you against infection. So if you're missing this protein called the CFTR, you can't release those red spheres, those anions, and they get trapped inside the cell. So like we had predicted with the iron, we thought, well, there must be a driving force for that bicarbonate to be released. It just can't get out because of the blockade that's happened with the loss of the protein. So if we could find a small molecule, now one that could transport the bicarbonate, perhaps if we added it to the apical side, you could release the bicarbonate and restore the function in the airway. And so again, now we were in a situation of trying to find such a molecule. Again, it did not come easily. We worked for almost a decade to find a molecule that could do this, in this case. Lots of mechanism studies along the way, lots of different discoveries, and finally we found one that looks really, really exciting, okay? This molecule is now called amphotericin B. This is another natural product, so it's a naturally occurring molecule. It was known to form ion channels for a very long time, but it was also thought to kill cells by forming those channels. And so to make a long story short, what my lab showed was that it does form channels, but those channels are not actually the primary mechanism of killing cells. It's something different that's killing them. And that allowed us to separate the channel activity from the undesired killing activity, and then use the channels for good, to do something positive. And so this was actually one of the key areas that we ran into a big challenge when we were trying to do this, that got us really excited about the synthesis methodology that I'll tell you about now. So bottom line, in trying to figure out how this molecule works, we needed to make it. And we also needed to make derivatives, so modify its structure and understand how it works. And this turned out to be the bottleneck in the whole program, is that the process of making these molecules was really, really slow. And therefore, we couldn't run nearly the number of experiments that we wanted. It was clear that there was no way we were going to get there if we didn't change something fast. The other thing that I want to highlight about this slide is this bottleneck represents not only trained chemists trying to make molecules quickly, but there's also a big bottleneck in terms of who gets to participate in the molecular innovation process. Because currently, despite 200 years of progress in our field, making molecules is still a highly specialized process that requires highly trained experts. And so to us, that represents a real loss of opportunity, right? Because if only a very select group of people can participate in the discovery process, just think about all the ideas we're missing out on because of those barriers to participation. And so one of the things we really want to do is figure out, how do we shatter these barriers towards molecular innovation and get lots of different people into the tent so we can get the benefit, right, of the kind of outsider effect, encouraging everybody to think differently and think creatively about how to solve problems. So with that goal in mind, we said, okay, can we figure out a better way to make molecules, and can it be something that's easier to access for everyone? So in that vein, we got really inspired by this kind of building block-based construction idea. So as many of you might know, the kind of industrial revolution is largely connected to this modularization of the making process. So that is, people started to make things from building blocks, and that systematization and modularization made it easier and easier to make stuff and to make lots of them and also to invent stuff, because you could make stuff quickly, tear it apart, and make stuff again. And this went everywhere from, you know, pulley blocks on ships to cars to buildings to space stations. And if you need any more evidence for this, if you just give a kid a bucket of Lego bricks and watch them go to town, it's amazing how powerful, you know, kind of Lego-like construction can be for, you know, turning someone's imagination loose. So with this idea in mind, we started to ask, why can't we do the same thing, but on the molecular scale? You know, could we develop Lego kits for molecules, for example, so that anybody could kind of participate in this process? So this was really the vision we came up with of this might be what the future of chemistry could look like, is you know, a pick and build model in which Lego bricks that are actually just little pieces of molecules could be snapped together to make new structures and thereby discover new function. And specifically, the way we did that was we took a reaction called the Suzuki-Miura coupling. It's a particular reaction that brings two carbon atoms and stitches them together. And we figured out a way to do that reaction kind of over and over and over again. So you take these building blocks that have, we call it a halide and a boronic acid, so two different functional groups attached to two different sides, and we then were able to use this bifunctional reagent as like the linchpin. So you can imagine like a train car that has a acceptor and a donor end, and then you just put the train cars together, right, to make the train. It's the same thing. We're going to make molecules like train cars, okay, or Lego bricks, where you just snap them together one at a time. And so the way we did that is we figured out a way to turn one end of this building block off, so it's like a protection for that piece, and that allows us to selectively couple these ends together, and then we remove that blocking group to make a new reactive unit, and then we do another coupling, and it's just kind of a repeat the same process over and over again. So you snap them together, you take off the protection, you snap it together, take off the protection, and repeat, repeat, repeat. And so my student Eric Gillis figured out how to do this, and thereby allowed this to be an iterative process. And this really helped us break through in terms of this Lego-like construction. And so this slide just shows some of now the hundreds and hundreds of molecules that we and many others have made all over the world using this Lego-like approach. And the thing I want to highlight is in each case, you see these colors. Those are the three different building blocks, for example, the blue, the red, and the green, that were then snapped together using the same reaction over and over again. So it's turned out to be a very general, very powerful way to make molecules. And then we built a machine. So analogous to the way machines can put other things together on the macroscopic scale and other types of molecules, we were able to make these kind of, you know, potential drug-like molecules using this new type of machine. And the beauty here is that it's so simple. You just put the cartridges on the machine, you press start, and then the entire process is automated. So you don't have to be a specialist in the actual chemistry of coupling in order to be able to now start to imagine how you might make new structures, because the machine becomes your partner in that process. And so we also spun a company out based on this. It's called Revolution Medicines. It's actually a very exciting story. There are more than 100 people now actually in a company out in California. They've got a drug that's in the clinic to try to help treat cancer, and they're doing great. So it's been a very exciting way to think about how a technology can lead to a translation into societal impact as well. Okay, so with that, back to the story about amphotericin, is that we were able to use this kind of Lego-like chemistry to make derivatives of amphotericin. So we changed the structure, and by doing that, we understood something for the first time. And that is, as I mentioned, the channels that we really care about, they do form, but that's not what's killing the cells. What we discovered is that the killing effect happens because amphotericin also forms this large aggregate or sponge-like structure on the surface of the cell, and it pulls out this green part, which is this cholesterol molecule or ergosterol in yeast, and it thereby removes that from the membrane, and that's how it kills. And so because we understood that, we were able to separate these two effects, okay? So we're able to change the system so that we only get the channels and we don't get the undesired effects, and that's what allowed us to move this forward. And so with that insight, we were able to now look back into yeast to try to understand whether this could work in the first place as a model system, and we got this really exciting result in which amphotericin was able to recover the growth of yeast that were otherwise unable to grow. So this is normal yeast, and you can see them growing on the plate just fine. This is yeast where we knock out their ion channel and they can't grow at all. And then we added amphotericin and we got full recovery of the yeast growth. So this was very encouraging. We then were able to take this and start working on comparing it to human-based epithelia from the lung that we could grow in the lab. We used as a control this compound. It was actually a recently clinically approved drug, which for certain types of the CFTR protein that are present, but they're like a little bit misfolded, it helps them fold properly and helps them work better. And so this was actually a really exciting success story that happened in the meantime in the clinic. And so we were able to compare our effect or our results to this molecule. This one can only work in very certain number of patients, whereas our approach could work in all the patients. So it was a very exciting way for us to get kind of validation, but also be able to reach beyond what this molecule could do. And so with that, we were able to show now in epithelia from the airway of people with CF who donated their cells, and we grew the cells in culture. We were able to see that you can get increase in the bicarbonate secretion. So this is an assay where we're measuring the amount of bicarbonate that's flowing across the airway. Here's where you start. And then when you add the clinically approved drug that we know makes people better, we get this nice increase. And when we now add the amphotericin, which is this different mechanism that we're trying to show that causes its own channel to form, you can see we get the same increase. Okay, so the fact that the green and the yellow look very similar is really exciting because it says that through a different mechanism, we're getting the same increase in bicarbonate secretion. And that causes the pH or the acidity of the airway to also get modified in a way that becomes more basic in both cases. And that's considered to be a really good sign for improving the physiology. So this was very encouraging result. We also showed in different types of lung epithelia from people that now can't respond to that other approach, we still see a nice increase in the bicarb flow and in the pH, suggesting that our approach might work for everybody with CF, which was really, really exciting result. So with that, we were able to then team up with a group at Iowa who was able to help us get access to epithelia from very courageous patients who donated their lungs when they had to get a lung transplant. And so then we could take those lung tissue and actually do experiments in the lab. And we saw really exciting results in which the ASL pH goes up. And we can see that and compared to derivatives that we made, this one that doesn't form channels. So we see this really nice specific effect. We also saw the viscosity of their airway surface go down, which is a good thing. So viscous surface means you can't clear out the infection. And so we decrease the viscosity. It makes it easier for this fluid to flow. And then most importantly, we saw the antibacterial activity go up. So this is directly counting, how much of the bacterium you can kill using the airway surface. And we see a really nice improvement caused by the amphotericin. And so with that momentum, we were able to move forward and check whether this could work in animals. And so the best model for cystic fibrosis in animals is in piglets. And so we were able to take pigs with cystic fibrosis, and you create a small opening in their trachea, and you can add the molecule directly to their airway. And in 4 out of the 4 pigs that we tested, we saw this really nice recovery of their ASL pH, which was very encouraging that this might actually work in people. And so I'm very excited to share with you a very recent result. We teamed up with an extraordinary young woman named Emily Kramer-Golenkoff, who has CF, and she is unhappy with the treatments that are available to her. And so to her credit, she started her own foundation. She calls it Emily's Entourage. She's raised millions of dollars to fund CF research. And we shared our results with her, and she decided to fund our clinical trial. So this is all compliments of Emily. The next study that I'm going to tell you about, she teamed up with my students, and we were able to do this study in people now with cystic fibrosis at the University of Iowa. 8 individuals who were unable to respond at the time to this other medicine I told you about. And the goal here, we added it to their nose, and you can measure whether or not you can change the electrical potential in their nose. And if you can, this is a good sign that it might work in their lungs. Okay, so it's a first look at whether or not this might be effective. And I'm very excited to tell you that 7 out of 8 of the people that we tested showed a really nice reduction in their nasal potential difference. And this was very similar to that which had been seen with that other drug I told you about in different people, giving us a really exciting first look, and actually the first demonstration ever that this might actually work in people. So we haven't published this yet, but I'm excited to share it with you today. Okay, so as we like to do, once we get to a certain point, we go ahead and spin out a company to translate. So we did this again. We just started a new company called Cystic Medicines, which stands for Cystic Fibrosis Prosthetics. And this company, we're in the process of trying to get it funded and translate this into clinical impact. Okay, Charlie, do I have a few more minutes to talk about Shield?
This is Lisa. You have about 23 minutes.
Perfect. Okay. So the other thing that we've been working on at the College of Medicine that I'm really excited to share with you is, as many, of course, are keenly aware, we're in a very challenging time right now, and the COVID pandemic has really, I think, challenged us as society to figure out, how can we keep doing all of the critically important things that we do as a community and do so as safely as possible. And so the College of Medicine is rightly looked to for leadership in thinking about how to do this here at the University of Illinois. And we also love to see ourselves as exemplars. As I think many of you know, our entire theme is innovation, right? We want to take challenges, refuse to accept that they cannot be solved, and then decide to innovate and do something hopefully that's very disruptive, that has high leverage, that can ultimately make a big impact. And so with COVID-19, the university looked to us and said, can you figure out a way to help us reopen the University of Illinois as safely as possible? And is there a way to think about testing for SARS-CoV-2, the virus that causes COVID-19? Is there a way for us to use testing to help us reopen campus and do so safely? And so when we were asked to do this, we took this challenge on, fully aware of how difficult this was going to be, but emboldened by the innovative spirit, okay, that pervades our culture of innovation, as Ruby Mendenhall likes to describe it, which I love. Let's say we can do this, okay? We can find a way to try to make a difference. And so the College of Medicine has taken this on. I've been very happy to have a chance to lead an extraordinary team focused on standing up and strategically deploying COVID-19 testing as part of our campus reopen effort. So I think the first thing that we realized when we were asked to really take on this challenge is that, look, this is all about health, right? We want to think about creating a healthy community that can reopen as safely as possible. And so we came up with this concept of a shield, right? So it's all about creating a protective barrier so that we can reopen in the context of the pandemic. And the second thing we realized is that there is no silver bullet. There's not one thing that we could do that's all of a sudden going to allow a community to feel confident that it can reopen. This really requires a multidimensional kind of synergistic strategy where testing clearly needs to play a major role, but testing alone is not going to be enough. So we decided to go after this in a very comprehensive way, in which we've got a kind of three-part strategy. We call it our target, test, and tell. The idea of target is, who to test and when to test them and how often to repeat it. The test has to be massively scalable in order to pull this off. And then the third part is the communication side. How do you get those results out to people as fast as possible? And how do we figure out who may have been exposed to the virus and therefore empower them to make smart choices, okay, about what to do next? So we took this comprehensive approach, and I'll tell you a lot more about the different components. The next thing we realized is that the tools and technologies that existed as of a couple months ago were not going to be good enough. So we realized we were going to have to innovate if we were going to pull this off. And the cool part is that's what we love to do in the first place, okay? So at Carle, Illinois, when we say innovation is required, we step up and say that's what we do best, okay? And so we decided to take this on fully aware that, we're going to have to innovate to make this happen. And then the last piece is we realized that no matter how, powerful of a program we could come up with, it's all going to be about individual personal responsibility, right? So everyone's going to have to do their part to make this happen. And so we really wanted to emphasize from the outset, this is about forming a strategic partnership between technology and people so that, everybody does their part and really figures out how to contribute to making a community safe. All of that we meant to capture by making this big I, okay? So this is the Illinois approach to protecting the community. Innovation is right at the front, and I can do this, right? This is something I need to do my part to make a difference, okay? So we put a lot of thought into this logo, and it's meant to try to capture all of those things. All right, so to jump right in and let you know the three different parts that we're talking about, again, target, test, and tell. So on the target side, this is where data science becomes really interesting and powerful. So rather than just randomly test everybody or have to test everybody every day, or you can imagine ways to do this that aren't very strategic, we said, what if we could develop an algorithm that can utilize the likelihood that any individual might be exposed, and then use that to help guide us towards prioritizing our testing deployment so that we get the most positive impact out of the resources that we have. Okay, so we call this a prioritized risk-guided testing strategy that really focuses on this kind of individualized risk exposure. And I'll talk about that in a little bit more detail. This is actually a really cool opportunity for thinking about, for example, artificial intelligence and frontier data science and, computer modeling, et cetera. And so it's a really cool kind of frontier data science problem to figure out how to do that. The second piece is we knew we were going to need a really effective test that was easy, cheap, scalable, and there was nothing like that that was available. So again, we decided to innovate, and I'll tell you about a brand new COVID test. We're calling it the iCOVID test, which is now based on saliva as opposed to the nasopharyngeal swab, and it skips the need to isolate the RNA. Okay, so the researchers here at Illinois and the Carl Illinois College of Medicine, first in the world to be able to show you could directly go from saliva to PCR. And we think this is hopefully going to have a huge impact, not just for us, but globally for how to get a handle on the COVID pandemic. And then the last piece is the tell part. So here it's all about getting the word out, all right, fast and effectively. And so we talk about exposure notifications. How do we know if we've been exposed to someone who had the virus? How do we do contact tracing with the Champaign-Urbana Public Health District in a way that is very fast and effective to understanding once someone's identified as having been positive for the virus, all the other people they may have interacted with. And then ultimately also, how do we communicate support for people's health? Because it's a scary piece of information to learn if you've just tested positive for COVID-19. How do we deploy resources very effectively so people rightly feel that a big bear hug comes around them and that they're given the right healthcare to help themselves get through this very challenging time, both their physical health as well as their mental health. And if, unfortunately they do get sick, how do we make sure they have really good access to the healthcare that they need? Okay, so these are all the different pieces of the puzzle. And as I mentioned, there's no silver bullet. One of these by itself isn't going to get us there. But if we pulled these three pieces together and took frontier science and technology and innovation, we felt like we had a chance to really do something special. And we like to say, this is 2020, not 1918. So we got to go on offense and take frontier science and technology and try to get ourselves in a position to reopen safely. Okay, so that's our approach. So I'll just tell you a few of the stories. It's been an amazing adventure. We have an extraordinary team working together on this. Charlie Simpson, who you met at the outset, is actually one of the leaders also on the team trying to help us figure out all the different communications angles associated with this. Jessica Breitbarth, also associated with the College of Medicine, has been absolute champion of figuring out how to strategically engage with communities and help fund this whole project and all these different aspects. And then I'll tell you a couple of the stories on the science side of groups that are working hard to innovate and make all this possible. Okay, so the first story comes from the labs of Rebecca Smith and Nigel Goldenfeld and Sergey Maslov. These are the extraordinary group of students and postdocs that they were able to deploy to work on this problem of figuring out how to target the COVID-19 testing in a way that would be most effective. And so what the students and the team came up with is this very exciting kind of new way to do this. We're calling this the kind of risk score guided prioritized testing algorithm. It's just a long way of saying we try to test in ways that make the most sense, okay, based on people's exposure. And so the idea is, let's say everyone's asked to fill out a survey. And your survey, you say, how many classes are you taking? How many people are in each of those classes? Are they live or are they online? What is your housing situation? Do you live with other folks in your dorm, et cetera? Do you have a work schedule? Do you do other non-university activities, et cetera, et cetera? And from all of this, you learn information that allows you to calculate an individual's likelihood based on their daily experiences that they could be exposed, okay, to people who have tested positive. And from that information, you can have an algorithm that figures out kind of randomly, but in a risk-guided way on a daily basis, test lots of different people and get lots of different data and hopefully get out in front of figuring out if someone tests positive and thereby stop the spread of the virus. Okay, so this is the algorithm that's been created, and this then taps in to help guide us in terms of how we deploy the COVID-19 testing on a daily basis. Okay, so the second piece was the testing part. And one of the other really cool parts of this story is that we had a very interesting opportunity at Illinois because we have a fantastic veterinary diagnostic lab that's really one of the world leaders in doing molecular testing for animals. And in fact, many of you may have heard that back in February, there was a tiger at the Bronx Zoo that was diagnosed with COVID-19. That was done here, actually, at the University of Illinois. It was a researcher by the name of Lei Wang, who literally wrote the book on coronaviruses in animals. And he was able to help the Bronx Zoo figure out that their tiger, in fact, had COVID. So we were able to then team up with Lei, as well as Tim Phan and Paul Hergenrother. Paul's also a member of the Illinois College of Medicine, and so therefore a great connection here. And they have a very exciting program, Tim and Paul, where they try to develop drugs for pets, and they translate those to people. So they've actually, for example, developed a cancer drug in dogs that now is being tested in clinical trials at Chicago and Johns Hopkins and other places that's looking really, really promising. So they've called this their Pets to People kind of program, and it's housed at the Institute for Genomic Biology here at Illinois. And so we teamed up with all of these folks and many others and say, could we translate the tiger COVID test to people? And so long story short, we've transformed our veterinary diagnostic lab into a human COVID-19 testing lab, and we're off to the races in terms of being able to have a facility to do the test. Now, the problem was the test still wasn't good enough, okay? So as many of you might know, the current version is a nasopharyngeal swab, followed by, you have to then isolate the virus, and then you can test using PCR, or polymerase chain reaction, as to whether or not someone has the RNA for the COVID in their nasopharyngeal cavity. The problem with that test is that it's really cumbersome. It's actually very unpleasant. It's slow, it requires a healthcare worker. It's very expensive. The actual viral isolation is slow and costs a lot of money, and therefore the whole process is slow and expensive and not scalable. So we wanted to find a way to do something different that could actually address this. And again, this is where we just kind of decided to innovate. It's actually a pretty exciting thing to do. You say, I don't know how I'm going to solve the problem, but we're going to make sure we do something different. And we just jumped in feet first. We teamed up, launched kind of like a Manhattan Project style effort over the last 6 to 7 weeks with an extraordinary team to go after this problem and try to get it to a saliva-based test to be a lot more efficient. So we had a really nice kind of head start from the literature. This was a study that got reported in April by a group at Yale. Even more sensitive than you could out of the nasopharyngeal swab. Okay, so this was a very encouraging result, suggesting that saliva was a potential medium for testing, but also very interestingly, saliva is the way we spread COVID-19. It's actually thought to be droplets as we speak or sing or cough or sneeze. And so it really actually probably is the medium that matters, right? So if you're gonna wanna understand whether someone is infectious, saliva gives you exactly that, that information about their viral load in the transport medium that's causing them to infect others. So we got really excited about this, even though it was only one study. We said, okay, maybe saliva could be a very powerful way to go. The problem with the Yale system is that they still had to isolate the virus, okay? And that still is a very expensive, very slow process. So we got excited about the question, could we skip the viral isolation step and go directly from saliva to the PCR machine? And this would be a huge increase in efficiency and lower the cost, et cetera. And so we built this extraordinary team of students and postdocs. You get the theme. Whenever there's a major important thing to happen, we pull teams of students together and empower them to do the innovation aspects. And so this is Diana Renoa, Fadi Analgi, Robin Holland, Kelsey Green, and Enrique Valera. And they formed an amazing kind of all-out full court blitz, working day and night to go after this. And they came up with a really exciting solution. This was work also led by Paul Hergenrother in collaboration with Tim Phan, Chris Brook, Rasha Bashir, and my lab. And so the idea was, here's the standard method where you've got the nasopharyngeal swab, the isolation, and then the PCR. There's a lot of bottlenecks in terms of how to do that. So it had been shown you could go from the saliva to your isolation and then the PCR. You know, could we skip straight from saliva to PCR and thereby make this much more efficient? And I'm very excited to tell you now, after a long story short, is that this actually has been discovered. So the group found, by looking at a whole bunch of different temperatures and times and buffers and additives, et cetera, a full matrix of different possibilities, this remarkable protocol that's super simple. So you just put saliva in a cheap tube that you can get, hundreds of thousands of them for very low cost. You simply heat the saliva for 30 minutes at this specific temperature. You add a specific buffer that was discovered through this kind of matrix of tries, and they directly show that the PCR reaction works, okay? So they probably tried about, I don't know, 10,000 different possible things, and this is the one that worked, okay? So really, again, the power of fearless failure, right? And lots of different shots on goal, and eventually you find something, and we just published this paper online as a preprint to shows all the data if you're interested of how we got there. We also developed an antibody test. This is the royal we. Preeti Sharma is an amazing postdoc in the Kranz lab, developed a way to do this to test for antibodies based on a report out of Mount Sinai. There's also now that report has come out in the Nature Medicine article shown here. Okay, I'll just close by saying the last piece is to bring all this together for this tell component. And the data shows that if you do kind of app-based contact tracing, you can get a certain effect. If you do human-based contact tracing, you get a certain effect. But if you put them together, it's the most effective way to stop the spread. And so we've actually teamed up with a group that's developed a new app called Rockwire, which is an app on your phone that can actually, using very privacy secure but very effective Bluetooth technology, it can tell you whether you've been exposed to somebody else who's had a positive test for COVID-19 without telling you who they are, where they were, or when you got exposed. So it's fully privacy protected, but it's a very powerful way to enable people to make smart choices about what to do next. Like if you've gotten an exposure, you can go quickly get a saliva test and find out if you're positive. We also have the Champaign-Urbana Public Health District doing contact tracing, and now we're working on merging these two to make them synergistic. The last piece is that we've teamed up with the OSF Healthcare to be able to provide immediate healthcare to anyone who tests positive. And this can be anything from just digital communication all the way to a kind of care package that shows up at your dorm that allows you to check your temperature and monitor your symptoms, and all the way to, if you want, directly connect to healthcare workers who can help you get the care that you need. So it's a very comprehensive, kind of very powerful program that will be available to anybody who tests positive. Okay, so to pull it all together, the last slide is we've asked everyone now to do your part. So as I mentioned at the beginning, this is all about personal responsibility driven empowerment. I think that's the concept. So we say everybody needs to get tested and participate in these programs, but also do the simple things. Of all the great technology we have, it turns out wearing a mask, staying 6 feet apart, and washing your hands regularly are going to be just as, if not more important than all of these other things. If we do all these things together, we have the real chance to make our community safer and thereby reopen in a way that hopefully allows us all to thrive despite the challenging situation we're in. Okay, with that, I'll just thank the extraordinary group of young people that I have the privilege of working with on a daily basis in my lab. It's their really creative and courageous discoveries that have driven everything I've told you about. We also have a really great opportunity to work with a lot of different funding sources and collaborators. Thanks so much for joining us again today. I guess I'll just conclude and tell you that Carl, Illinois is just an amazing place where we get to do this type of research. I've shown you just a tiny bit of the amazing collection of stuff that's going on across our entire college, and thanks so much for giving us a chance to describe. I'll be happy to answer any questions, Charlie, if that's possible.
Yeah, we have about 5 minutes for questions. So if you have any questions for Dr. Burke, feel free to put them in the chat and he can address them. I will also put our reach email in the chat. So if you have questions after this webcast that you think about, we can get it to Dr. Burke or the right person to answer that. Dr. Burke, there was a question earlier, Carissa. I don't know, I think it was in reference to your CF slides, but asked about, just said, isn't that an antifungal? How does that make the channel? What's the mechanism?
Yeah, it's a great question. The answer is yes. That's absolutely correct. So amphotericin has been used as an antifungal agent for more than half a century. It's actually the last line of defense for really tough fungal infections. And interestingly, it's got a big problem in terms of its toxicity. So patients and doctors rightly criticize it's called amphoterrible because of this really bad side effect. And it was known to form ion channels. And it was thought that that's how it kills the fungi, is that the channel activity was thought to be the mechanism of killing. And unfortunately, it was thought also that's how it kills human cells, was by forming channels. And if that were true, that would make it a very bad starting point for a molecular prosthetic, because you would have a toxic channel activity, and they're hard to imagine how you would use that to help increase physiology if you're deficient. What we learned, and again, it was about 10 years of work, finally discovered that mechanism is not correct. So amphotericin does form ion channels, and that's not how it kills yeast primarily. And it's also not how it kills human cells. It's this other mechanism, we call it the sponge-like effect, where it removes sterols from the membranes very quickly. And that actually allowed us to separate the channel activity from the antifungal and the toxicity to human cells and turn the channel into something that can restore physiology as opposed to destroying it. So yeah, it's a very interesting story, that a good example where diving into the basic science and understanding mechanism can open up opportunities for rationally taking advantage of function that wouldn't be obvious unless you understood it better. So thanks for the question.
Looks like we have another question. How do you, is it possible for undergrad students with a CS background to get involved in your lab?
Oh, awesome. Well, thank you for that question. Absolutely. So we have lots of collaborations with computer scientists. Nate Russell, who's a grad student in Jen Peng's lab in CS here at the University of Illinois, for example, sits in on our group meetings regularly, and we've teamed up with him to develop algorithms to figure out how to make molecules into Lego kits and do that systematically. And we're actually now working on a program with computer scientists to figure out how to get AI driving the automated synthesis machine, and then automate the testing, and then the data comes back to a computer, and then AI decides what to make next. So you call this kind of a self-driving laboratory. Just like you imagine a self-driving car, we're trying to get kind of fully autonomous molecular discovery driven through this closed loop type system. So we have all kinds of interesting computer science-based projects that interface with our chemistry goals. And yes, if someone's interested, please send me an email. We'd be really excited to talk to you.
And then we have, how do you choose the small molecules to screen that may have activity in treating disease?
Yeah, it's a really interesting question. How do you find what you're looking for? At least give yourself the chance to find what you're looking for. So we've actually gotten really excited about this yeast-based assay. So we're very much like yeast at the molecular scale. It's very interesting. Millions and millions and millions of years of evolution have still kept us quite close to yeast when it comes to the general hardwiring of how these things work. And in fact, so many of our proteins have a yeast homologue, meaning we can even see the protein that we came from a long time ago, the one that still exists in us. So what we do is we knock out the protein in yeast, and then we get a phenotype where the yeast can't grow. So then we look for molecules that cause the yeast to grow again. And thereby we're getting a readout on whether they're replacing the missing protein function in a very kind of holistic way. So we call this a phenotypic assay. That's how we discovered both the henochidiol and the amphotericin. So now we're thinking about a project where let's take all the human diseases that are caused by a missing protein, knock out the homologue in yeast, and then screen for molecules that cause those yeast to grow again. And each one of those hits could be a takeoff point for making a new medicine. So that's a really interesting kind of new way to think about discovering these molecular prosthetics that we're really excited about.
So we're right at 3 o'clock, but Dr. Burke, there's one last question, if you want to go ahead and answer that. For the algorithm that analyzes risk for an individual, it will take into account where you go and who you're in contact with. Will it also take into account an individual's health in general, like any problems one may have?
Okay, very interesting question. So it turns out the way it's designed, it's just about your activities, period. Because the idea behind it is that based on your daily activities, there's a somewhat increased or decreased chance that you're going to be exposed, largely just based on how many people you probably interact with on a daily basis. So we don't rely only on that. The idea is that we're going to do lots and lots of testing, and it just somewhat kind of targets in a way that if you have lots and lots of exposure on a daily basis to other people, you're probably going to get more pings on your phone that it's your turn to get tested than others. But you'll still get pinged even if you have very low, kind of risk exposure, because we of course don't want to over rely on this. So it's really just meant to kind of guide, and it's all based on your activity. Whether or not we would take into account your individual health, no, because obviously at the end of the day, whether or not you have certain health conditions, we don't know or not, whether it makes you more or less likely to get COVID. So we don't really think about that as one of the criteria. It's more about your daily activities and how many people you come into contact with.
Great. Okay, thank you, Dr. Burke, and thank you everybody for joining us today. We appreciate your time.
Thanks so much. Great. Thanks so much for joining us.